This presentation covers the organs of digestion and their functions. Digestion is the process in which food is broken down into its building block molecules for absorption. Here is a diagram of the organs of digestion. It is important that you be able to identify these parts and know what they do. The digestive system is also known as the GI tract or the gastrointestinal tract. It's approximately 9 meters or 10 feet long. There are two types of organs in the digestive system. There are primary and secondary or accessory. Primary organs are those organs through which food passes. Secondary or accessory organs are those organs that play a role in digestion, but food does not pass through them. Remember that the primary organs of digestion are those organs through which food passes. They include the mouth, the pharynx or throat, the esophagus, the stomach, the small intestine, and the large intestine. Accessory organs of digestion are those organs that aid in digestion, but food does not pass through them. They include the teeth, the tongue, the salivary glands, the pancreas, the gallbladder, and the liver. Most of the digestive system is composed of 4 different tissue layers. The mucosa is the innermost layer. It's composed of stratified squamous epithelium, and its purpose is protection. The submucosa lies just to the outside of that and is composed of loose connective tissue, and it contains the blood supply. The muscularis is just to the outside of the submucosa and is composed of smooth muscle, and its job is to propel food. And finally, the outermost layer is composed of simple squamous epithelium and loose connective tissue, and it is known as the serosa. Its purpose is also protection. There is one organ in the digestive tract that has no serosa. It's the esophagus. Here again is a list of those 4 layers of the digestive system. To the right is a microscopic slide showing the location of each of those layers. Take a look at another microscopic slide and see if you can label the 4 layers. The key is on the left. Let's go through each of the organs of digestion, beginning with the mouth, also known as the oral cavity. In the oral cavity, you have both mechanical and chemical digestion. In the mouth, you also have 4 different kinds of teeth. The incisors, which cut food, the canines, which tear food, premolars or bicuspids that grind food, and molars that crush food. You can see in the diagram on the right, each of these 4 types of teeth is labeled. There are 2 dental formulas that you will need to recognize and understand. Dental formulas tell the number of each type of tooth in 1/4 of the mouth. For an adult, that dental formula is 2-1-2-3, indicating there are 2 incisors, 1 canine, 2 premolars, and 3 molars in 1/4 of the mouth. So that indicates either half of the top or half of the bottom. In a child, the dental formula is 2-1-0-2, meaning in 1/4 of the mouth they have 2 incisors, 1 canine, no premolars, and 2 molars. So in order to calculate how many teeth there are, you would need to multiply that number by 4. So in the case of an adult, 2 incisors times 4 means an adult has 8 incisors. 1 canine times 4 means an adult has 4 canines. 2 premolars times 4 means an adult has 8 premolars, and 3 molars times 4 means an adult has 12 molars. Saliva plays an important role in the process of digestion because it's 99.5% water. It helps to soften the food and make it move more easily through the digestive tract. In addition, saliva contains salts and organic molecules like mucus that helps to lubricate, lysozyme, which is bactericidal, and amylase, which digests starch. There are 2 types of digestion that occur in the oral cavity: chemical digestion, and we'll be covering that later on in this lecture, and mechanical digestion, also known as mastication. This is when food is mechanically broken down and a bolus is formed. A bolus is a ball-like mixture that contains saliva and food, and it forms in the mouth during the process of chewing. Once this bolus is formed, deglutition or swallowing must occur. There are actually 3 stages of deglutition or swallowing. The first is the voluntary phase, and this is when the bolus moves from the mouth or the oral cavity into the pharynx or the throat. The pharyngeal phase is when the bolus moves into the esophagus, and the esophageal phase is when the bolus moves into the stomach. If you view this slide in slide presentation mode, you'll be able to see peristalsis moving the bolus through the esophagus. At the top of the esophagus is the upper esophageal sphincter, and at the bottom is the lower esophageal sphincter, also known as the cardiac sphincter because it lies near the heart. It takes about 4 to 8 seconds to move the bolus from the top to the bottom of the esophagus. This wave-like movement again is called peristalsis. Overall, the esophagus is about 10 inches in length. At the entrance to your stomach is a valve, which is a ring of muscle called the lower esophageal sphincter, or the LES. Normally, the LES closes as soon as food passes through it. If the LES does not close all the way, or if it opens too often, acid that's produced by your stomach can move up into your esophagus, and this can cause symptoms like burning chest pain called heartburn. If acid reflux symptoms happen more than 2 times a week, you have acid reflux disease, also known as gastroesophageal reflux disease, or GERD. There are common risk factors for acid reflux disease, like eating large meals or lying down right after a meal, being overweight, eating a heavy meal and lying on your back or bending over at the waist, snacking close to bedtime, eating certain foods such as citrus, tomato, chocolate, mint, garlic, spicy foods, or fatty foods, drinking certain beverages such as alcohol or carbonated drinks, smoking, being pregnant, taking aspirin, ibuprofen, and certain muscle relaxers all put you at greater risk for acid reflux disease. Here is a diagram of the stomach. Be sure you know all of these parts. The stomach is a highly distensible organ. That means it's very stretchable. The stomach has 2 functions, and that is to store food and release it slowly, and the mechanical and chemical digestion of food. At the bottom of the stomach is a valve called the pyloric sphincter that controls the movement of food from the stomach into the small intestine. In the stomach are folds known as rugae. The function of rugae is to provide more surface area and to expand as needed. There are 2 types of digestion that occur in the stomach. They are mechanical and chemical. Mechanical digestion is the process of churning the food, and this is possible because there's a muscular layer known as the muscularis that's thick and allows this to happen. There are 3 layers of muscle in the muscularis. They are circular muscles, longitudinal muscles, and oblique muscles. And again, the presence of these 3 different kinds of layers and their crisscrossing allow you to churn your food and do it very efficiently. This diagram shows you the 3 muscular layers: the longitudinal muscles, the circular muscles, and the oblique muscles. The second type of digestion that occurs in the stomach is chemical digestion. The stomach secretes gastric juices that are colorless and watery in consistency and have a pH between about 1.4 and 1.6. Those digestive juices come from mucus cells, which secrete mucus, zymogenic cells that secrete pepsinogen, enteroendocrine cells that secrete gastrin, and parietal cells that secrete HCl. It's this HCl that gives these gastric juices such a low pH. HCl, or hydrochloric acid, has 3 very important jobs. It ruptures cells, it activates pepsinogen, changing it from an inactive form to an active form, and it kills bacteria. There's a mucus lining in the stomach that will protect it from HCl and the active pepsin and prevent it from being digested. There's little absorption that occurs in the stomach except for aspirin and ethyl alcohol. The 3 phases of gastric secretion are cephalic, gastric, and intestinal, and we'll look at each of these. Let's begin with cephalic. The cephalic phase occurs before food enters the stomach. Triggers for this phase are aroma, taste, sight, and the thought of food. And there are certain factors that will dampen this response, like depression and lack of appetite. This phase is controlled by the vagus nerve. The second phase of gastric secretion is the gastric phase, and it begins when food reaches the stomach. Triggers for this phase include stomach distension or stretching, the presence of proteins and peptides in the stomach, a pH down to 2, caffeine, and moderate concentrations of alcohol. One of the triggers for the gastric phase is stomach distension. When food reaches the stomach, it begins to stretch. This stimulates the vagus nerve, which then sends an impulse to the gastric glands and causes the release of gastric juices. The vagus nerve is important, but it's less important than the effect that the hormone gastrin has on this process. When there are proteins, peptides, a low pH, caffeine, or alcohol in the stomach, it causes the release of the hormone gastrin. Remember, this is more important than the effect of the vagus nerve. Gastrin causes two things to happen. One is it will send a message to the gastric glands and cause the release of gastric juices. The second function of gastrin is to increase motility in the stomach. And remember, this is the churning of the food within the stomach. Both of these things are important in digestion. The two primary things that can interfere with the gastric phase are a pH of 2 or less and emotional upset. The third phase of gastric secretion is the intestinal phase. The intestinal phase begins when the stomach empties into the duodenum. There are certain factors that control this gastric emptying. They are the hormone gastrin, which promotes this process, GIP, which is gastric inhibitory peptide that inhibits the process, and the enterogastric reflex, which also inhibits the process. The enterogastric reflex is when the pyloric sphincter tightens and food cannot move out. We've already talked about gastrin, and we said that it promotes gastric emptying. Let's look next at GIP, gastric inhibitory polypeptide, which inhibits gastric emptying. GIP is produced by the enteroendocrine cells of the intestinal mucosa. Its production is triggered by acid, peptides, and fat in the duodenum, and the most important of those three is the presence of fat. All three of these will slow gastric emptying. That's why this is inhibitory. The targets of GIP are the muscularis of the stomach, it will decrease motility, and the gastric glands of the stomach, which will decrease their secretion. The last factor we want to consider in gastric emptying is the enterogastric reflex, which is controlled by the vagus nerve. There are several triggers for this reflex. They include distension or stretching of the duodenum, acid in the duodenum, peptides in the duodenum, and fat in the duodenum. And the most important is the presence of fat. The effects of this reflex are to decrease gastric motility and a strong contraction of the pyloric sphincter. Be sure that you understand these questions and their answers. What happens to the rate of gastric emptying with a high-fat or protein meal? And the answer is there is delayed gastric emptying. Why? Because fats and proteins trigger GIP and the enterogastric reflex. What happens to the rate of gastric emptying after Chinese vegetable stir-fry? The answer is you soon feel hungry because there is rapid gastric emptying. And the reason it is so rapid is because there's little protein or fat in the meal. Because food mixes with gastric juices in the stomach, by the time it enters the small intestine, it is changed from a bolus to a soupy mixture known as chyme. Small intestine is about 21 feet long and it has 3 segments. They are the duodenum, which is 10 inches long, the jejunum, which is 8 feet long, and the ileum, which is 12 feet long. Because it has several modifications, the small intestine is the major site of chemical digestion and absorption. Those modifications include plicae circularis, villi, and the microvilli. All 3 of these increase surface area. The large intestine begins where the small intestine ends. The major function of the large intestine is to absorb water from the remaining indigestible food matter and transmit the useless waste material from the body. In addition, the large intestine contains bacteria that produce vitamin K and vitamin B12. Accessory organs of digestion, or organs that play a role in digestion but food does not pass through them, are the pancreas, gallbladder, and liver. All 3 of these dump digestive enzymes into the small intestine. Here is a diagram showing all 3 accessory organs. This slide shows you a microscopic view of the pancreas, including an islet of Langerhans. The pancreas secretes 2 very important solutions. They are a bicarbonate solution and a solution of digestive enzymes. Because the solution coming from the stomach to the small intestine is highly acidic, and the duodenum does not have a protective covering like the stomach, it's important to neutralize this solution. So the presence of acid chyme in the duodenum causes the endocrine cells of the intestinal mucosa to release the hormone secretin. Secretin stimulates acinar cells to release a bicarbonate solution, or HCO3-. This neutralizes the acidic chyme. The second solution released from the pancreas is a solution of digestive enzymes. The presence of fats and peptides in the duodenum causes the endocrine cells of the intestinal mucosa to release the hormone CCK, or cholecystokinin. This hormone then stimulates the acinar cells and the gallbladder to release digestive enzymes. The digestive enzymes of the pancreas include trypsin, chymotrypsin, and carboxypeptidase, all of which are proteolytic enzymes, or enzymes that digest proteins. In addition, it also contains pancreatic lipase, which digests lipids, pancreatic amylase, which digests starch, ribonuclease, which digests RNA, and deoxyribonuclease, which digests DNA. The liver is another accessory organ, and it produces bile, it detoxifies, it stores glycogen and vitamins, it produces plasma proteins, it removes old worn-out red blood cells, and it converts nutrients from one form to another. Here is a macroscopic view of the lobes of the liver. The liver produces bile, which is composed of water, cholesterol, salts, lecithin, which is a phospholipid, biopigments like bilirubin. Its main function is the emulsification of fats. This is not the digestion of fats, but emulsification means making big fat drops into smaller fat droplets that can then be digested. This slide shows you how bile works. You have a big fat drop that bile will emulsify or break into small fat droplets. It's then the job of pancreatic lipase to digest it into its components, or its building blocks of glycerol and 3 fatty acids. The functions of the gallbladder are to store bile and to concentrate bile. Let's take a look at carbohydrate digestion and absorption. Carbohydrate digestion begins in the mouth with salivary amylase. There are actually 2 important enzymes in carbohydrate digestion. They include salivary amylase, which is produced in the mouth, and pancreatic amylase, which is produced in the pancreas. This diagram is an overall view of the digestion of polysaccharides. Salivary and pancreatic amylase act on polysaccharides, or starch, and break them down into disaccharides. Those disaccharides are sucrose, maltose, and lactose. The disaccharides are then broken down by the enzymes known as disaccaridases, sucrase, maltase, and lactase, into building blocks of monosaccharides, glucose, galactose, and fructose. You should be able to answer these questions about polysaccharide or carbohydrate digestion. Where does carbohydrate digestion begin? And the answer is in the mouth. Name 2 important enzymes that break down polysaccharides, and they are salivary amylase and pancreatic amylase. Name the 3 disaccharides: sucrose, maltose, and lactose, and what enzymes break each down. And they are the enzymes sucrase, maltase, and lactase. Whenever you see the ending ASE, it tells you it's an enzyme. When you see the ending OSE, it tells you it's a sugar. Name the 3 monosaccharides, and they are glucose, galactose, and fructose. Once we digest carbohydrates, they then need to be absorbed. The next few slides will cover how that happens. Before we talk about actual carbohydrate absorption, let me orient you to this slide. Down the center are 2 bold lines, and they represent a capillary. To the left of that, you're looking inside the intestines, and those green cells are epithelial cells of the mucosa of the intestines. These building blocks have to cross those epithelial cells to get into the capillary. And let's see how they do it. Remember that the breakdown products, the building blocks of carbohydrate digestion, are glucose, galactose, and fructose. In order for glucose to cross the epithelial cells of the mucosa of the intestines, they need ATP. This is called active transport. It's the same with galactose. It also requires ATP to cross the intestinal cell. In the case of fructose, it does not require ATP, but instead requires a carrier molecule, and this is called facilitated diffusion. Now let's look at the processes of protein digestion and absorption. Proteins are digested into shorter peptides by the enzymes pepsin, trypsin, chymotrypsin, and carboxypeptidase. These peptides then are broken into amino acids by dipeptidase and aminopeptidase. The last slide showed how proteins are digested. Here are some things you should know about digestive enzymes. Where are pepsin, trypsin, chymotrypsin, and carboxypeptidase formed? And the answer is pepsin in the stomach and all the others in the pancreas. In what form are these enzymes secreted? And it is in their inactive form. And how is each activated? On the next slide, we'll take a look at that. On the left side of this slide are the enzymes in their inactive forms. On the right side are their active forms. In each case, be sure you know what activates the enzyme. Pepsinogen, the inactive form, becomes active in the presence of hydrochloric acid. Trypsinogen, inactive, is activated by enterokinase and becomes trypsin. Chymotrypsinogen is activated by trypsin and becomes chymotrypsin. And procarboxypeptidase is activated by trypsin as well and becomes the active form carboxypeptidase. Why are these enzymes in their inactive forms? And the reason is we want them only in their active forms to digest when there is food present. We just looked at how proteins are digested. Let's look next at how they are absorbed. You saw this same slide when we looked at carbohydrate absorption. We're now looking at protein absorption. Down the center, remember, is the capillary. To the left, you're looking inside the intestines, and those green cells are the epithelial cells of the mucosa of the intestine. The breakdown products of protein digestion are amino acids, peptides, and dipeptides, and all three require ATP to cross the membrane and get into the capillary. Again, since we're using ATP, this is known as active transport. Last, we'll look at lipid digestion and absorption. Remember, there are a couple of steps in lipid digestion. First, the large fat drops are emulsified into small fat droplets by bile, which acts as a detergent. The small fat droplets are then broken down into their building blocks of glycerol and fatty acids by pancreatic lipase. You can see the structure of glycerol and the fatty acids below. Note that short-chain fatty acids are between 2 and 10 carbon atoms long, and long-chain fatty acids are between 12 and 18 carbon atoms long. Let's look next at how these lipid breakdown products are absorbed. Here is the same diagram you saw with carbohydrate and protein absorption. Down the center is the capillary, to the left is inside the intestine, and the green cells are the epithelial cells of the mucosa of the intestine. If you take a look, the breakdown products of fat digestion were short-chain fatty acids and long-chain fatty acids. Both will diffuse across the epithelial cell of the mucosa of the intestine without ATP and without a carrier molecule. And it's because likes dissolve likes. They are fatty in nature, as are the membranes. This type of diffusion is known as simple diffusion. The term diverticuli refers to sac-like pouches. So diverticulosis is a condition of the presence of abnormal sac-like pouches in the intestinal wall. It usually affects those who are over 50 years of age, and it's most common in those with low-fiber diets. The problem is pressure builds up and these outpouchings occur. Diverticulosis is painless unless there are bacteria. When bacteria invade, the condition is then called diverticulitis, which means an inflammation of the diverticuli. It's important to have a high-fiber diet and to avoid developing diverticulosis and possibly diverticulitis. In the past, there were certain foods you were told to avoid. That is now controversial. Those foods were any foods with seed like tomatoes, cucumbers, corn, strawberry, and nuts. Also, avoid low-fiber diets, and that is still the common belief. Diverticula occur throughout the gut, but by far the most common and the most important are those that occur in the colon. You can see these outpouchings or diverticuli in this diagram. You can see the pouches or diverticula in both of the pictures above. The one on the left is a gross picture of the colon. The one on the right is a radiologic picture of the colon. Here is a picture of a normal colon with no outpouchings or diverticula. Compare this view with the previous view of a normal colon. You can see the pouches or the diverticula clearly. Compare these pictures of diverticulosis and diverticulitis. The one on the left is diverticulosis, which shows you the outpouchings. The one on the right is diverticulitis, which shows you the outpouchings with bacteria or an infection. The ending itis, itis, means an inflammation of. About 25% of people with acute diverticulitis will develop a complication. These complications include things like an abscess, which occurs when pus collects in the pouch, a blockage in the colon or small intestine caused by scarring, an abnormal passageway known as a fistula between sections of the bowel or the bowel and the bladder, or peritonitis, which can occur if the infected or inflamed pouch ruptures.